Fellow. She leads the stem cell research at UCL at the Institute of Neurology, and she was also awarded the ALUK Early Career Researcher Award last year, which was very prestigious and very happy that she was acknowledged in that way. And Charlie's been working with her for 5 years now, I think, and last year he was awarded his own fellowship from Alzheimer Society to take forward his FAD research. So please come and looking forward to hearing.
Can everyone hear me? Is that projecting?
No, it's turned off now.
Oh, it's turned off. I'm a good scientist, honestly. How do I switch it on? Should I just take that off? Is that better?
Whoa!
Yeah.
Too much. So it's really nice to be back here and talk to you about some of our research again. So as Natalie mentioned, too much?
Yeah.
That's okay?
Yeah.
That's okay, good. So Natalie mentioned I spoke at this meeting in 2011, and it's really nice to see some familiar faces again. And at the time that I spoke about this research before, we were really just at the beginning and we hadn't even got started. So for me it's really nice to be back and share with you some of our progress of using cell models to try and understand familial Alzheimer's. So Charlie and I will share this session. I'll speak for the first half and just give a bit of an overview of the technology that we're using in the lab. And then Charlie will take over and share with you some of the results that we've had so far. So as I mentioned when I last spoke, we were at the beginning and it was just me and we hardly really started, but there's now a whole team of us. So the work that we'll talk about today is done by the people in this picture. We're all based over in the labs, just about 10 minutes walk away from here, and everyone's really using the same approach to try and understand Alzheimer's disease at the cellular level. So we are going to speak today about molecular neuroscience. I think scientists are quite fond of using complicated words to describe quite simple things. We'll try and present it in an understandable way, but both Charlie and I will be here for the rest of the day. So if anything's not clear, you can ask us at the end of the talk or get us in the lunch break. We'd be really happy to answer any questions. So when I say molecular neuroscience, what we really mean is that we're trying to understand the small stuff. So all of the work that we do in the lab is trying to understand the DNA and the cells, the neurons, that come together to make organs such as our brain that eventually come together to make us as people. And I showed this schematic because I think it illustrates nicely that if something starts to go wrong at the very smallest level, that the impact of that is felt at each increasing level of complexity. So if there's a change in a gene or your DNA, that can lead to cells not functioning properly, and that can eventually lead to diseases such as Alzheimer's disease. So by extension of this, what we believe is if we want to successfully intervene in diseases such as Alzheimer's with drugs, we really want to target this scheme as early as possible. So what we're really trying to do in the lab is understand what are the very first things that go wrong in cells of somebody with Alzheimer's disease. So a lot of our knowledge around this has come from studying brains and looking at what happens in the brain tissue when somebody's lived with the disease. Now, of course, it's really quite tricky to study diseases that affect the human brain because it's not very accessible to us during life. We can't readily take a piece of someone's brain while they're still healthy and well. So a lot of the work we've kind of built our understanding on has happened by studying the brain at autopsy. And we heard in the first session that in Alzheimer's there is the build-up of proteins in the brain that wouldn't normally be there. The two particular proteins are one called tau, which is shown on this bottom image, and then these A-beta plaques shown on the top image. Now, the difficulty of studying brain tissue is we can see what's happened, but it doesn't give us a sequence of events. It doesn't tell us what order things are happening in and what's coming first. And this is really where, as scientists, genetics helps us to start to piece together a sequence of events. So we know there are 3 genes that are linked to familial Alzheimer's disease, APP, presenilin-1 and presenilin-2. And these are all associated with the production of the plaques. So what this tells us is the plaques happen first, and then that initiates a sequence of events which leads to tangles and eventual neurodegeneration. So once we have genetic information, we can use that to try and understand the cell biology of disease. So I think there's quite a common analogy that our DNA is kind of a blueprint for what makes us. I prefer to think of it almost as a parts list. So we know that all of our genes are the individual parts that make us, but without having context, we don't know what they do. So if you think about a parts list to make a car, we know that you need a steering wheel, you need brakes, you need seats, but it doesn't tell you what would happen if you took the brakes out. We need the context of the whole car. So what we do in the lab when we know that a gene is linked to disease is we use that gene to create models that we can study in the lab. And I've put some commonly used lab models in this image, so we can have something as simple as proteins in a tube, or we can have models such as mouse models, such as fruit fly models, where we can take the genetic change that we know is linked to disease and put it into one of these systems and look at the effects of having that gene present. Now, of course, as I've already mentioned, all of these models have their strengths and their limitations. And that's why I've got the picture of a jigsaw, because we need to use them all to piece that information together. But as I've already mentioned, one big limitation we have is that up until recently we've not been able to study human brain cells in the lab. The reason that's a limitation, and again I'm sure most of you don't need to be told this, but the human brain is quite different to a mouse brain. Certainly quite different to a fly brain for most of us. So the human brain is really to my mind, the most complicated system in the known universe. We don't understand how it works fully under normal circumstances, so it's difficult without that base knowledge to understand what's going wrong in disease. But this really is where stem cells can help us. Okay, so again, I'm sure that everyone has heard of stem cells in one way or another, but I'll just give a little bit of background that hopefully will help to make the rest of the talk clearer. So when we talk about stem cells, for the purposes of our work, we're talking about cells that have two specific properties. So the first property is self-renewal. This means a stem cell will divide to make two identical cells, each of which will divide to make two identical cells, and so on and so on. And that process of self-renewal will continue indefinitely until we make use of the second property of stem cells, which is the property of pluripotency. So this is, again, scientists use a fancy word to say something quite simple. It means most potential. So what that is really saying is that a stem cell is a blank slate, and within that cell there's the potential to become any cell type of the human body, given the correct signals. And that of course includes neurons, the cell type of the brain that is affected in Alzheimer's. And the technology we use that we'll talk to you about was really developed by groups who wanted new ways to make stem cells. So a lot of stem cell research comes from cells that are derived from early stage embryos before that embryo is developed. And several groups couldn't use that approach because it was restricted in certain countries due to kind of ethical concerns. So what they did was something quite remarkable, and they turned really a kind of a dogma of biology on its head. So we can see here we've got the stem cell with the kind of arrows indicating the specialization of those cells. And for a long time it was thought that that was a one-way street. So if a stem cell became a skin cell or a muscle cell, that process was thought to be irreversible. But we've known now for the past 10 years that that's not the case. We can change the identity of cells from being one cell type to another. And that's what we do in the lab. It was first described using skin cells, which are much easier to access than brain cells from living patients. We can ask participants to donate a piece of skin, and in the lab we can change the identity of those skin cells and essentially reset their clock. We can take them back in time to a point when they were unspecialized. So we're inducing the property of pluripotency. So these cells are called iPS cells. What that means is because we've induced that property of pluripotency, we can then turn them again into the cell type that is affected in disease, and that includes neurons. So what it means for us is we can ask people who know that they carry an Alzheimer's associated gene if they would be willing to donate a piece of skin to us. We can then use that piece of skin to make the stem cells and the brain cells in the lab. So for the first time, what we have been using in the lab for the past number of years are brain cells which we have grown from research participants to understand what's going wrong in those cells from people with Alzheimer's disease. So I'll just show you a little bit of what this looks like. When someone agrees to participate in this study, they have a small skin biopsy taken from the upper arm in the clinic. This comes to us in the lab, and it comes to us in a fully anonymised way, so we don't know anything about the individuals who donate the samples other than what genetic change that they have, their year of birth and their sex. That's the information that we get. In the lab, you can see in the top panel what we do is dissect that skin biopsy just into smaller pieces. And then the way that we grow cells, again, is quite simple. We have them in an incubator that keeps them at 37 degrees, so at body temperature. And they're sat in a sort of pink fluid which contains everything that our cells get from food. We just buy it off the shelf in a bottle that we give to our cells. Actually, Charlie's nipping over to the lab to feed the cells during the lunch break because they're very, just like us, we need to feed them every day, and that includes weekends. So if you see him nip out, that's where he's gone. And then after a few days, what you can see, this is what one of the pieces of skin looks like under the microscope, and you can see that the skin cells are starting to grow out from that piece of the biopsy. And then the final image here is just showing that we can actually freeze those cells and essentially hold them in suspended animation. So we can store the cells indefinitely. Whenever we want to work with a particular cell sample, we can just bring that up from the deep freeze and it will thaw and grow. So starting from these skin cells, we then eventually make the brain cells, and I've just got a few images of what those look like before I hand over to Charlie. So these are some brain cells that we've grown in the lab. I hope you can see that okay. I think they look really beautiful. There's a clearer image on that screen if the light's interfering. So to me, they always look like a firework that's just exploded. I think they're really beautiful to look at. This structure is actually really important. When the brain develops, it develops in the first stage of development on a tube structure. And what we're looking at here is almost looking down on top of that tube in the red and blue. And then the green processes that are radiating out of that structure are the cell's axons. So these are the long processes that allow the cells to talk to each other in the brain. And they're the processes that actually start to break down in Alzheimer's disease. We can actually eavesdrop on the cells talking. Again, I'll play it a few times, it might be better on that screen. What we've done here is we've put a dye on top of the cells that binds to calcium. Every time a neuron fires a signal or communicates with a neighbouring cell, it has a fluctuation in its calcium levels. So if we just run that video, that's what you can see is happening there. So even though they look still and static in the dish, they're actually chatting away to each other. And of course what we can now look at is how that communication is affected in cells from people who carry an Alzheimer's gene. And then my final slide before I hand over, these cells are grown in a single sheet on top of a layer of plastic, but we're now able to, thanks to the technology progressing quite rapidly, we're also able to now grow 3D tissue-based models. And this is from what we call a cerebral organoid. It's quite commonly referred to in the press as a mini brain. These are, I think that's a stretch, but it's a catchy term. These are models that are about the size of a lentil, and they grow in the lab, and what we've done here is taken a slice through one of those and stained it for different cell types. And the reason that I showed this is every colour on the screen is a different cell type, and you can see that they are occupying different spaces within the model. So this is moving us closer towards what a more realistic model of the brain, where we have spatial compartments of different cells. So that's just an overview of the technology that we're using, and I'll now hand over to Charlie, who will tell us a little bit about what understanding it's given us.
Okay, thank you Selina, thank you to Natalie and all the organisers for today. My name's Charlie and I'm going to be just talking about some of the results from one of the projects that we've been conducting here at UCL with our colleagues in Sweden. And just to say that the research is really into the basic biology behind familial Alzheimer's disease, and just to really stress that I think this is really important in understanding what's really going on behind the disease in order to generate the next generation of drugs, and a better understanding of really what's happening in the brain is going to help us going forward. So as Selina introduced already, amyloid beta is the main component of the plaques that are found in the brain. It's really this relationship between the Alzheimer's genes that leads to this amyloid beta production. However, that amyloid beta doesn't come in just one fragment, it comes in several fragments, which is pictured down here. And when the enzyme cuts the amyloid precursor protein, the APP gene, it doesn't just produce one fragment. In a Pac-Man type way, it chomps and chomps, and we get these two different pathways of how amyloid beta is produced, leading to smaller and smaller fragments as we go to the right hand side down here. And we can see that amyloid beta that's 40 amino acids in length, so amyloid beta 40, this is the most abundant type of amyloid in the brain, and is thought to be not harmful to brain cells. Whereas amyloid beta that's 42 or 43 in length, amyloid beta 42, is thought to be more toxic to brain cells. And it's really this relationship between the Alzheimer's genes that makes this amyloid beta that we're trying to understand, and the effect of the Alzheimer's genes on this processing, on how amyloid beta is produced. Okay, so when we take our stem cell lines with the either from non-AD individuals or research participants with gene changes in APP or presenilin, each one of these bars in the graph shows a different cell line, and when we turn those into neurons or brain cells in a dish, we can start to see the amyloid that is produced by these cells. And we're lucky enough to have one of the largest panels of stem cells that we know of, and we can start to understand these changes in how amyloid is made in the cells. So when we study amyloid beta 42, or the toxic one, relative to the more abundant one, 40, we can see that the AD lines produce more of the toxic amyloid beta, and this is thought to be because of the way that the Alzheimer's genes are affecting this processing of how amyloid beta is made. Just to say that we see very similar results in the 2D neurons and also the mini brains as well, so this is really due to the gene changes affecting how amyloid beta is made. However, it's not just as simple as that. When we look at some of the other fragments that are made in this pathway, we can see that different gene changes have different effects on how amyloid is made. And so when we look at amyloid beta 38 and 40, what this is showing us is the difference, the first choice, whether it goes on the pathway on the top to make amyloid beta or the pathway on the bottom towards amyloid beta 40. We can see that gene changes in the APP gene have a big effect on which pathway is chosen. So amyloid beta is actually shuffled along the top pathway, and that's the reason why we're seeing more of the 42, more of the toxic fragment. This doesn't seem to be the case in this measure for gene changes within presenilin 1. However, when we look at some of the other measures, such as amyloid beta 42 to 38, so the red arrow at the top shows us that we're measuring how effective this final cut is of the Pac-Man as it's going down this pathway, we can see that the gene changes in presenilin 1 have a great influence on how much this final cut happens. And so this is the reason why we're seeing more of the 42, more of the toxic amyloid produced in these cells. So the reason why these cells are making more amyloid beta 42 isn't all the same, and I'll just mention a couple of the reasons why, and the reasons why this genetic change is affecting amyloid beta production, but they're not all the same. So the gene changes in the APP gene seem to block the top pathway, and that's really because this amyloid beta 46 is changed by the mutation. So you can think of it as a roadblock on that top pathway, shuffling the cutting down the bottom pathway, and for that reason we're seeing more of the toxic 42. In contrast, when we look at the gene changes in the presenilin 1, we can see, for example, if I use the arrow, we can start to see less of the enzyme being produced in these cells. And one example of this is, you can see in this particular line, this is an immature form of the enzyme, and it's not producing the active form of the enzyme. So for that reason, there's less of presenilin 1 enzyme. And so when we look at these 3 lines here, you can imagine that less of the enzyme around to cut up the amyloid leads to less efficient processing towards the smaller fragments, and so that's why we're seeing more of the toxic species produced amyloid beta 42. So I'll just finish by saying why is all this important, and I think what's really important here is that we can really see the very early changes in the cell, so the underlying effects that gene changes leading to familial Alzheimer's disease have on brain cells, and this is really for the first time using this brain cells in a dish technology. But I think the results from this first project really shows that not all patients or not all gene changes have the same effect. And I think this is important for 2 reasons. Firstly, that the different disease presentations or the different forms of familial Alzheimer's disease might start to be explained because of some of these differences. But also this will be really important going forward in drug trials and things like that. So some patients might respond to some drugs differently to others. So I think it's really important to understand how these different effects will have an effect on the disease. And I'll just finish by saying we're taking this study forward, for example, looking at the other cell types in the brain, not just the neurons. So the support cells, which are shown here in the bottom, making these beautiful cells in a dish, and also the immune cells. I'm taking work forward with Selina looking at the immune cells in the brain, and really trying to understand how these early gene changes can affect the brain, leading to amyloid beta and familial Alzheimer's disease in time. And so I think a greater understanding is really going to help us start to reverse some of these changes and help us generate the next generation of drugs. So with that, I'll hand back to Selina.
Thank you. So we'll just summarise, and then both of us are here for questions, and we're both around in the lunch break. But hopefully we've shown you today that one of the barriers to developing new successful treatments is that the human brain's been inaccessible to us. But thanks to these new advances in stem cell technology, we can now grow human brain cells in the lab. It's revolutionised neuroscience as a whole. And for us, it's starting to give us unique insight into the individual changes in people with familial Alzheimer's disease. We've not had this before because when we've used kind of mouse models and other cellular models, they typically are built around one change. And what we're seeing now is that we can't always extrapolate that to be universally applicable. So looking ahead, although it is quite far away from now, I think it's good to be ambitious, and this could help us with personalised medicine. That's already happening in the cancer field, and I hope that we'll get there one day too. And finally, and most importantly, we wouldn't be able to do any of this without research participants. We've been really humbled by how willing people are to contribute samples to our study. So thanks to you all for your attention, and we'll be very happy to answer any questions.
Oh yeah, I don't need to mention one more thing I forgot to mention.
I'd also point out that the other Charlie Murphy is here, and as well as growing samples from individuals, we've grown some of Charlie's cells. Charlie's an artist who's kind of responded to how she feels about having her brain grown in a dish. And as we're taking that work forward, she'd be really keen to have some patient voices and research participant voices about how you all feel about this type of technology. I've also grown a bit of Nick. I've got Nick in the deep freeze, which is...
Oh, you're used to the space.
Yeah, so I don't know, some people maybe at a previous meeting where I mentioned something that I think is really one of the most exciting developments in terms of finding an effective treatment, which is there are lots and lots of what we call neurodegenerative diseases, where brain cells or nerve cells die, which is essentially the problem in Alzheimer's disease. And I think I mentioned before, but for those people who weren't here, there's a childhood neurodegenerative disease which is genetic, and instead of the degenerating cells being particularly around brain function, it's around the nerves and support muscles, so nerves and muscles. So these children who are born with a gene defect die at a very early age, depending upon what particular gene. And some of them, the effect on the muscles is so bad that they never ever it can be so bad they never ever get the strength to lift their heads off cots. And it's, this is the most heartbreaking stuff, because children at a very early age. And very, very recently, treatment trial, I'll tell you about what the treatment was, had to be stopped early. Most of the time, those people who've been in treatment studies know that one half of the people, one get a drug and the other half don't, and nobody knows who's it's all called, what's called blinded, so it's all, neither the doctors nor the people in the trial know what they're in. And you have lots of tests, and I'm sure many of you here have had tests which are either biological measures, or they're things like cognitive testing, and then try and see whether there's a difference. The trial had to be stopped early because it was so obvious that the kids on the trial were not just doing better than the kids not getting the treatment. The children who are having active therapy, they weren't just doing better than the children who were not getting the treatment, but they were catching up their normal brothers and sisters. So instead of just dying, they were starting to learn to walk and ride tricycles, things that just would, in the history of that disease, had never happened before. You got the gene, this is what happened to you, didn't get the gene, you were normal. And now suddenly that was changed. And the therapy that was used was something called ASOs. And what that is recognizing, using a treatment drug that is like the other half of a zipper that matches exactly the chunk of the DNA or the genetic material RNA that where the genetic defect is. And it's often being talked about as gene silencing. So it just sticks to that defect, and that can't have its effects, so it can't make the abnormal protein, which then makes all the bad consequences. And the really exciting news is that we have just started such a program, still at the very early stages, to do that with familial Alzheimer's disease. And the first step will be, and Selina and I have been on, and Charlie have been on others, on teleconferences just in the last few weeks with a small company that is going to make us a number of these to test. And how we will test them, because of course you can't say, oh, this might work, let's put it in the people. We'll start with these stem cells, which is just amazing, because if we could start seeing the cells that Selina and Charlie make behave like they don't have the gene if we put in this silencer, that is a really powerful signal that should be taken forward. And there's a lot of testing to go for before you go into humans, but it's really exciting that there's a potential that what we saw of a genetic disease for the first time ever in neurodegeneration. I mean, cure is a very big word, but getting close to what one might have thought, which is I didn't believe that we would see. So that is a very specific and urgent way that we will be using these cells, and it's really exciting. Anyway, I've hijacked the end of Selina and Charlie's talk, but just to mention that, because I think that's really makes real what we're doing with these cells. In addition to the beautiful pictures that Charlie will be doing. Okay, so questions to you. Do you want? Is it questions for
any
time?
questions for Selina and Charlie, or again, there'll be opportunities to ask to speak to them over lunch. But does anyone have any questions now?
Too mindful.
Okay, well, we'll take a lunch break then, but please do feel free to come up with them. I know they love